we are going to detect protein in urine. So we will perform heat acetic acid test and sulfosalicylic acid test. Okay, the principle behind these two tests is protein denaturation of protein structures. Okay, so take a test tube, fill it 2/3 with this sample. Fill the test tube up to 2/3. Then we will heat the which part? Lower part, upper part, whole test tube? Upper part. We will heat the upper part of this test tube until there is a white coagulum appears. Okay. Now, bas, ঠিক আছে. Now hold it with a test tube holder. Here is the burner. Someone heat the upper 1/3 part. Upper 1/3. Why we heat only the upper 1/3 part? To keep the lower part as control. Okay, the sample is transparent now. After heating, it will be whitish, cloudy appearance will be there if protein is present. হয়েছে আসছে whether there is any cloudy appearance. হয়েছে? Now add 2 drops of glacial acetic acid. Why we are adding glacial acetic acid? Why we add glacial acetic acid? If the cloudiness is because of protein, 2 drops, okay? So the cloudiness will remain as it is even after adding 1% glacial acetic acid, and if it is because of phosphates or carbonates, it will be cleared. Now reheat it. After adding glacial acetic acid, we will reheat the upper part again. The cloudiness is same. So this confirms the presence of protein in this sample. সবাই বুঝেছে? Clear? Perform the salicylic acid test. Take another test tube. অন্য কেউ করো এবার, তুই কর. And take 3 ml of sample. 3 ml of sample. You have to take 3 ml only. 3 ml is sufficient. হয়ে গেছে তোদের? আচ্ছা, এটা কি টেস্ট করেছিস? Heat and acetic acid test to detect which abnormal constituent will be detected? Protein. Why we do heat test? What is the principle behind heat acetic acid test? Denaturation of protein. And why we add the glacial acetic acid? So protein coagulum will be dissolved by glacial acetic acid. Yes or no? Acha. So what are the substance that can give rise to cloudiness, white flocculum after heating, but will be dissolved after adding glacial acetic acid? So protein will not be dissolved. So which other substance will give white flocculum and then it will be dissolved by adding glacial acetic acid? Phosphates or carbonates. Okay, so phosphates and carbonates, if present in the solution, they can also give white flocculum on heating, and they will be dissolved after adding glacial acetic acid or then heating. Okay, but if the coagulum stays as it is, it confirms the presence of protein. Clear though? And this test is salicylic acid, sulfosalicylic acid test, and the principle behind this is same. That is denaturation of protein. So we don't heat this sample, we just add the salicylic acid. How much? 5 drops is enough. Okay, in 3 ml of sample. 3 ml of sample, we will add the salicylic acid and there will be a cloudiness, overall cloudiness. ইউরিনের abnormal constituent কি করেছিলি তোরা? বেনেডিক্টস করা হয়েছিল? কিচ্ছু ক্লাস করেনি নাকি? ক্লাসে ছিলিস না? এই এদিকে সরে দাঁড়া, সবাইকে দেখতে পাই যেন। বল কি টেস্ট করা হয়েছিল আগের দিন? বেনেডিক্টস করা হয়েছিল। কেন করা হয়েছিল বেনেডিক্ট? টু detect which abnormal constituent? Reducing sugar. Okay, and today we will perform test to detect proteins in urine. And what is the test we will perform today? Heat acetic acid and sulfosalicylic acid. Okay, so these two tests are done on the principle of, what is the principle behind these tests? Denaturation of protein. The denaturation of protein পড়া হয়ে গেছে কিন্তু ক্লাসে। Okay, so secondary, tertiary and quaternary structure, they are denatured and only the primary structure is maintained. Okay, and heat is one of the denaturing element, and sulfosalicylic acid is also a denaturing agent. Okay, so what will we do? একজন আয় test tube নে, fill the test tube with the sample up to 2/3. পেছনে বার্নার জ্বলছে, একটু সাবধান। হ্যাঁ, 2/3 of the test tube should be filled. ওইভাবে না, ওটাকে আগে বাল্বটাকে ডিফ্লেট করতে হয়। এয়ারটা আসছে তার কারণ এখানে কিছু ফ্লোকুলাম ইয়ে হয়ে আছে। যে হয়েছে, ফিল হলো, এরপর এটাকে দে টেস্ট টিউবটা। হয়েছে 2/3। ব্যাস, নাও এবার ওটাকে একটা টেস্ট টিউব হোল্ডার দিয়ে ফিক্স করো। কেউ কিন্তু খালি হাতে টেস্ট টিউব নিয়ে হিট করবে না। Okay, always fix your test tube with test tube holders and then heat. ওখানে আছে বার্নার। Heat the upper 1/3 part, upper part of the test tube only. And why we heat only the upper part? Why not the whole test tube, or why not the lower half?
Coagulum will form in the upper part.
Yes, coagulum will be formed and the lower part will be taken as control to compare. To compare with the transparent lower part, we can compare it easily. Clear? So which test is this? Heat acetic acid, and this sulfosalicylic acid. So both the test is dependent on which principle? What is the principle behind these two tests? Denaturation of protein. Okay, denaturation of protein. In case of heat acetic acid, heat is the denaturing agent, and in case of sulfosalicylic acid, it's the sulfosalicylic acid itself is the denaturing agent. Okay, and why are we adding glacial acetic acid? হিট দিয়ে তো পেয়ে গেছিলাম ফ্লোকুলেশন, তো why are we adding glacial acetic acid? হয়েছে, কোয়াগুলাম এসছে। নাও add 2 drops of glacial acetic acid, just 2 drops of glacial acetic acid. Why? Any other substance that can give coagulation on heating? That is phosphate and carbonates. Okay, phosphates and carbonates, they can also give such white coagulum on heating. And to conform proteins, we add glacial acetic acid. If it is phosphate or carbonate, it will be dissolved. Okay, 3 ml of sample. স্যাম্পল তো ছিলই, ওটা আবার ওটা ডিফ্লেট করলি কেন? ওটা দেখ স্যাম্পলই ছিল। টেক 3 ml of sample, approx 3 ml। দেখ না এখানে 4 এ আছে, আপ টু 7 তুই স্যাম্পলটা ড্রপ কর। আপার মেনিসকাসটা 4 থেকে 7 অবধি আসলে ছেড়ে দে। দ্যাটস ইট, হয়ে গেছে, ছেড়ে দে। আচ্ছা, দিস ইজ 3 ml, around 3 ml. Now add 5 drops of sulfosalicylic acid. বাকিরা দেখতে পাচ্ছিস? 5 drops। হ্যাঁ, হয়েছে। মিক্স করারও কিছু দরকার নেই, একটা ক্লাউডিনেস অ্যাপিয়ার করেছে, দেখতে পেয়েছিস? দেখিয়ে দে তোর বন্ধুদেরকে। হয়েছে, ক্লিয়ার হয়েছে তো? তো দিস কনফার্মস প্রেজেন্স অফ প্রোটিন। ওকে,
Okay, so first we are going to do our protein detection in urine, and we are going to do heat and acetic acid test and your sulfosalic acid test. Okay. So what are you supposed to do? You are supposed to fill up with the sample first. Okay. Look how it is done. First of all, look how it is done. Take about 10 ml of your solution into a test tube. See, you have to take 10 ml only. Okay. You have to take 10 ml only because this tube has two parts. One is the control part and one is the test part. Okay. Can the technicians light up the Bunsen burner, please? আরেকটু নিয়ে নিবি, আরেকটু নিয়ে নিবি, সুবিধা হবে। হ্যাঁ, 2/3 part of the fill ওটাই বাস। You have to take a few drops of acetic acid. You have to take a few.
So we will heat the upper half of the test tube.
You are supposed to heat it till the bubbles start forming. Okay. Once the bubbles start forming.
Can you see the cloudiness? Okay. Now we will add 2 drops of glacial acetic acid. Okay. And we have to boil it again.
So why did we put glacial acetic acid to it? We got the flocculation first hand, right? So why do you need to buy another reagent? Dissolve হয়ে গেলে কি হতো? Yes, so this was done. Carbonates. So basically phosphates and carbonates will give you false positive in case it is raised in concentration. It will give you a false positive test if it is raised in concentration. That is why, in order to ensure this is not a false positive but a true positive, we are adding glacial acetic acid to the flocculation. Okay? Happy? Very good. Now please proceed with the sulfosalic acid part.
And in this solution, in the sample, we will add 5 drops of salicylic acid. See, cloudiness at that? This is also denaturation, that is also denaturation. That's it. Any one of you perform the heat acetic acid test and any one of you perform the salicylic acid test.
That's more than enough. No happens. Leave, we do not have excess of sample. Okay, that's more than enough. Now hold it with the burner. Hold it with the test tube holder. Heat the upper part till you get bubbles. Usually. Usually, once the bubbles start forming, you can see it's flocculating below the point where the bubble is being generated. Okay.
Is there no active when we get the flocculation afterwards?
No. Add 2 drops of glacial acetic acid, and that will do. One, two. Very good. You might shake it a little. Just shake it a little. Yeah. Happy? Hit it. Why did you shake it? So that you get a uniform distribution of glacial acetic acid. Okay. No, no more. No more. Wash the test tube. Deload the components and wash the test tube. Okay. Someone else. Dara sulfosalic acid koreni. Hey, Dika, do the sulfosalic acid test. Help your friend out. Just take 3 ml. Okay. Just take 3 ml. You do not need that much. Just take 3 ml. Take one. You took 1 ml, right? I asked you to take 3 ml. It's not a hard and fast rule that you'll have to take 3 ml only. Okay? Take. Yeah, that's more than enough. Hello. Yeah, here is the sulfosalic acid. Hello. হ্যাঁ কিসের জন্য সেটা জিজ্ঞেস কর না বাড়িয়ে দেবো বাস আপাতত দুজন করুক অতটা তো নেই রিয়েজেন্ট নেক্সট টাইম আবার হবে নেক্সট টাইম পরের সপ্তাহে প্র্যাকটিক্যালি আবার রিপিট হবে হ্যাঁ সি ইউ ডু নট শেক ইট ওকে you do not shake it. If you shake it, then the flocculation will break and it will become homogeneous. Okay? So do not shake it. Make sure you do not shake it. You will see beautifully the pattern is being formed. Okay? So you leave it at that. Once you shake it, it becomes homogeneous. Once it becomes homogeneous, you will not be able to appreciate the flocculation process. Okay? So do not shake it. No, it will not be transparent color. It will become cloudy only. But here you will have a little bit cloudy, a little bit transparent. So it is easier for you to observe the flocculation process. If you shake it, that flocculation is going to break and it is going to be present in the entire test tube. Okay, and you see the protein itself, the sample itself is a little cloudy due to the presence of high amounts of protein. Then it becomes very difficult for you to appreciate whether it was like this only or whether it appeared after you put in the sulfosalic acid. Okay, that is why I am asking you, you do not shake it. You let the flocculation be. Okay, now just drop the contents, wash the test tubes and send the next step in, next people in. Happy? You have written your practicals?
heat and acid test. So heat acid test, why we do heat acetic acid test? Coagulum hobe. So protein will be denatured and we will find a cloudiness, right? So we will take sample. ঠিক আছে, 2/3 of the test tube is filled with sample. Now we will heat the upper 1/3 part and we will get a white coagulum or a cloudiness flocculum on the upper part. Okay. Can you see? The cloudiness appeared. Why we heat only the upper part? We use the lower part as control to compare the upper part and the lower part to see whether there is actually cloudiness or not. Okay, the lower part is transparent. হয়েছে? নিচের পার্টটা একদম clear, transparent, but the upper part there is cloudiness, and the cloudiness might be because of protein denaturation or any other substance on heating. They can give phosphate and carbonates. Phosphates and carbonates also can give white coagulum on heating. ঠিক আছে? So to confirm whether this is protein or not, we will now add glacial acetic acid. If this protein, so this is the confirmatory test. The adding of acetic acid, glacial acetic acid, confirms the presence of protein. হয়েছে? Clear? বেশি লাগবে না, একটু হলেই হবে। Okay, sample. Okay, and now we will add, and always keep your reagents closed. Okay, and how much salicylic acid? 2 ml. 5 drops only enough. Okay, see, the solution is clear, sample is clear. Now I am adding salicylic acid reagent. I added 5 drops. Okay, হয়েছে? Cloudiness. So what is the principle behind salicylic acid test or heat acetic acid test? I told you. What is the principle? হয়েছে? Cloudiness সবাই দেখতে পেয়েছে? Clear তো? Denaturation of protein. Principle is denaturation of protein. Okay, so denaturation can be done by heat or acids. So denaturing agent, sulfosalicylic acid is one of the denaturing agent, and glyceracetic acid is a no. Heat is a denaturing agent. Okay, clear? এবার তোদের মধ্যে থেকে তোরা কেউ একজন হিট অ্যাসিটিক অ্যাসিড পারফর্ম কর, একজন স্যালিসাইলিক পারফর্ম কর। ওকে, বাকিরা দেখ, আর একজন হিট কর, একজন গ্লেসিল অ্যাসিটিক অ্যাসিড অ্যাড কর। 2/3 of the test tube. গুড, হ্যাঁ ঠিক আছে, ভেরি গুড, ভালো হয়েছে। আর কেন এটা দুটো আলাদা পার্ট রাখা হয়? Why the only upper part is heated, not the lower part, or the full test tube is not heated? Why? To compare. Okay, the lower part we take as control part to compare whether the cloudiness is the proper cloudiness and the lower part is transparent or not. If the whole sample is cloudy, then we can't define. হয়েছে? গ্লেসিল অ্যাসিটিক অ্যাসিড অ্যাড করেছিস, অ্যাড করার পর রিহিট করেছিস, হয়েছে? এখনো আছে ফ্লোকুলেশন, তার মানে প্রোটিন প্রেজেন্ট। আর আরেকটা এর সাথে কোয়েশ্চেন যেটা করে সেটা হচ্ছে যে আর কোন সাবস্টেন্স থাকলে হিট দিলে ক্লাউডিনেস আসতে পারে? ফসফেট এন্ড কার্বনেটস। ওকে, কিন্তু তাহলে গ্লেসিল অ্যাসিটিক দেওয়ার পর ওটা ডিসলভ করে যায়, whereas প্রোটিন অ্যাজ ইট ইজ থেকে যায়।